initiative out of the Thank you. This is an initiative out of the research working group of the MCIA, and we hope it's valuable to you. I would like to ask that if there's any ideas for future workshops, please do put them in the chat function and we'll capture them. Just to let you know, if you didn't hear the little announcement, that we will be recording this session. We are very lucky today to have Professor Andrew McLachlan from the School of Pharmacy, University of Sydney, and Anastasia Sarayev of the Lambert Initiative with us today. I won't take too much time away from our presenters and from the opportunities for your questions. Just a couple of housekeeping points before we start. You have been muted as you come into this session. If you could please keep your microphones muted during the presentations. There'll be an opportunity for questions at the end. For questions, if you could please use the chat function at the bottom of your screen. Indicate you have a question so I can call on you, or enter your question there and I'll put it to the presenters for you. I urge you to take the opportunity to pick the brains of Andrew and Anastasia, who have a wealth of experience to share. Now I'd like to hand over to Andrew McLachlan, who will provide an overview of the ethical considerations in designing cannabis-based medicine trials, followed by Anastasia's presentation on the practical considerations of trial design, collating evidence, and the requirements around the investigator's brochure and investigational products. Okay, over to you, Andrew. Thank you.
Thanks very much, Melinda, and it's great to be on a program with Anastasia and her experience today, and hopefully we can address some of the questions you might have and also even point you in the direction of some of those answers. So today I'm coming to you from the land of the Gadigal people of the Eora Nation here on the Camperdown campus of the University of Sydney. And it's that important tradition that I acknowledge, and for any people of Aboriginal and Torres Strait Islander background, I acknowledge your and pay respect to you today as well. So my topic, as you can see, is to really provide a bit of an overview of some of the ethical considerations when it comes to generating evidence about cannabis-based medicines. And we're in a really exciting and important time at the moment about generating evidence for particular indications and also particular dose forms. And they're kind of my main themes that I'll talk about today. My email address is on the front there, and I'm very happy to respond to any questions after this session or, of course, during the session as well. Just by way of disclosure, I wanted to let you know that obviously I work for the University of Sydney. I have funding from a number of organisations, including the NHMRC. I've worked with a number of pharmaceutical companies in the sector, and also companies that are involved in the sponsoring cannabis-based medicines. I'm involved with the Lambert as an investigator and also part of their executive advisory board, and I've been involved with a few clinical trials in this space. One of the other things I'm involved in as well is a member of the Bellbury Human Research Ethics Committee, and part of my perspective today, of course, is some of that experience. So I'm drawing some of my comments today from a range of places, but of course, the views that I express today are my own, and I'm responsible for all of them. So what I'd like to talk about today is understand some of the key ethical principles involved in the design and conduct of clinical trials in this space. I'm going to take you back to some fundamentals, so I apologise for those people online who probably know this, but I'd like to start from a mutual base of understanding, if you like, and the importance of what we broadly call PICO. And I'll explain, I'm sure you know what PICO is, but it's about really the key questions that we're going to ask when it comes to designing our trial to generate the right answer and relevant evidence. I'd like to know about the essentials of investigating cannabis-based medicines, including aspects of quality and reliability that I know Anastasia will pick up on as well when it comes to information in the investigator brochure. Appreciate the importance of a study design and particularly in developing a protocol that is able to address some of the modifiable and non-modifiable risk factors that will be relevant to the study population. So if I take it right back to where ethics starts, of course, an ethics committee that might be considering an application is really informed by the 4 principles of medical ethics about beneficence, doing good, non-maleficence, not doing harm, about autonomy, respecting the individual and the need to provide informed consent, and of course justice, fairness, not only in the conduct of the study, but also perhaps continued access to a medicine after the trial, which is part of that story. Every human research ethics committee that's accredited, and the vast majority in Australia are, really adhere to the national statement of ethical conduct in human research. There's other resources too when it comes to specifically related to medical cannabis and information there, and of course particular guidance from the TGA around the conduct of clinical trials. So these key documents are really important, and I'm very happy to make a copy of my slides available for you to have a look at after this. Of course, the principles that we're really looking at here are quite broad when it comes to investigating any medicine, and that's the principle of the quality use of medicines, which does fit in our national medicines policy. I apologise for mentioning national medicines policy this early in the day. It's a very good sedative. But the reality is that these principles are really quite important. The first one, of course, is what is the best management option for a patient? This is exactly the decision-making process an ethics committee would look at. What is the best treatment? If it is a medicine, what is the most suitable and what is the current best option? And when we use those medicines, we do so safely and effectively. And doing that, of course, comes down to knowing more about what is the medicine itself. Is it a combination? Is it a full spectrum extract? Is it particular ingredients? What dose regimen? And of course, what dose form are very relevant to this idea of efficacy and safety. So, of course, I probably don't need to tell this group who have joined a meeting to learn more about clinical trials why we do clinical trials, but I have this slide, of course, just to remind us about when and why we do clinical trials. And it does come from the Clinical Trials Handbook, which is a useful resource, I think. I suppose the important bit is that it's the best form of evidence we have to guide not only aspects of, say, regulation of medicines and registration of medicines, but also to inform best practice and, of course, the quality use of medicines. So that idea of testing and monitoring the effect of a treatment on a larger number of people to ensure that there's benefit there and there's a manageable expectation of avoiding harm is really quite important. Efficacy and safety is, primarily the main goal here, and of course that would underpin perhaps an application to support a medicine for registration. It's the same standard that really needs to be met there. And interestingly, one of the themes that I'd like to highlight today, I'm sure will come out in the next presentation as well, is that evidence can be very specific to a cannabis medicine product, because it's such a broad definition when we use the term medical cannabis or cannabis-based medicine. We're talking about a whole range of possible constituents in different strengths, combinations, and of course the dose form. The other really important area is that medical cannabis or cannabis-based medicines will be used as either an adjunct to or an alternative to current treatment options. So in designing a trial, the relative efficacy and safety compared to current best treatment is also a very critical lens an ethics committee will look at. Again, this audience, I'm sure, doesn't need to know about the different phases of trials, but you would appreciate that the lens of an ethics committee is trying to establish, is this preclinical research that might support a first-in-human study? Is it a phase 2 study that's trying to demonstrate, does this treatment actually work? Phase 3, of course, we're trying to figure out how well does it work? Who is the right combination? What's the right combination of dose regimen and indication? A better understanding about safety in the pre-registration sense. And many studies are also phase 4, understanding an extension of indication or understanding more about safety and efficacy in practice. So that different lens is important. There's a really important paper that came out 2 years ago, led by Jennifer Martin, Professor Jenny Martin from Newcastle, but with a great team of very experienced researchers in this space. And you can see from the title, it's right in the sweet spot of what we're talking about today, about guidance for ethics committees, governance officers and researchers to think about this approach of how we take it on. And I have provided a copy of this paper to the organisers, but you can certainly find it online as well. And some of the things I might just step through, which really come out of this paper, are really quite important. So first of all, is a consideration about selecting the medical cannabis product for the trial. And if I think about my own experience in working with companies, in conducting research, and also in evaluating applications to ethics committees, defining and characterizing the product is absolutely critical. So knowing about the dose form itself, how it's prepared, and particularly if it adheres to good manufacturing practice standards, understanding some of the issues around access and guaranteed consistent supply has been very important for some of the trials that I've been associated with, given that many trials may go over many months and sometimes years. Having continued access to that particular therapeutic product is essential. And also knowing about, what we know about the product. And I won't talk more about this now, because I know Anastasia will pick this up when it comes to talking about the investigator brochure and whether a sponsor has that. Of course, in Australia, there's a clinical trials notification process as well. So one of the key questions is about accurate dosing and administration. So which cannabinoids, which combinations, what type of extract is involved? Are there many cannabinoids and predominantly CBD or THC focus? How is the medicine administered from a route of administration? And some evidence to justify the selection of the dose regimen and the dose form and route of administration relevant to the indication. And often that may involve information about pharmacokinetics for key constituents like CBD or THC. Appreciating the likelihood and monitoring for the frequency and severity of possible adverse reactions can be very important. This may be related and will be related to the nature of the product, if it's THC related or CBD predominant or a balanced formulation. We can expect a very different adverse effect profile. So safety data which underpins the use of that to inform, particularly phase 2 and phase 3 studies, phase 4 studies, becomes very important from a decision-making point around that beneficence and non-maleficence decision that an ethics committee will be weighing up. I'd also add in, as a pharmacist, I'm quite interested in drug interactions, and this is a really active area where more research is being done to try and better understand how CBD and THC particularly might interact with other medicines that someone is taking. And if you think about it, most of the trials will be done in a patient population who may be taking current best treatment, and of course, potential interactions needed to be identified there, which can of course lead to increased harm or perhaps even diminished response. So that becomes very important. Also then, of course, other things that people are ingesting, cigarette smoke, alcohol, and other perhaps even complementary medicines might be very important to avoid in designing the study as part of the exclusion criteria. So just to pick up on that point, a great article here from Jonathan Arnold, one of our colleagues at the Lambert Initiative and here in the Sydney Pharmacy School. A great resource in just articulating some of the characteristics that are relevant here. And you'll see, of course, for a THC predominant product that we can expect a very different spectrum of possible adverse effects compared to CBD alone. But drug interactions, you'll notice, are on both sides of the ledger. The informed consent process is a critical part of human research ethics evaluation and the conduct of any high quality trial. And one of the challenges in this space as well is, I suppose, the popular and general information about medical cannabis and what people assume about it or what they already know about it, or perhaps even are using cannabis, are now interested in medicinal cannabis type of trials. So we also know that many people who might be participating in some of these trials, based on the literature and also where we're seeing these medicines used, are really quite vulnerable. So they may have a life-limiting condition. They might be trying to control symptoms. They could be very vulnerable. Children, severe epilepsy, terminal phase of life. And of course, people are at that stage where they're willing to try anything to alleviate difficult symptoms. And that does create quite a challenge to make sure that people are fully informed before they consent to be involved in a trial where there's some question. The question really is about what is the role of a cannabis-based medicine in their care. A really important issue, and I mentioned this about justice in that framework of ethical decision-making, is will there be access to the medicine after the trial? So if you think, for example, about a clinical trial, phase 3 clinical trial that might provide benefit for reducing seizure frequency in a child, might improve quality of life if we take it to that degree, and trials have demonstrated this to some extent. You know, will the person who participated in that trial, will they be able to get access to the medicine in an affordable way at the end of the trial? So sometimes there might only be a clinical trial supply, and an ethics committee will often really prosecute that with the investigator. How will people be able to get this if it helps them? And one solution to that is to have an open label extension where people stay in the trial, they continue to get access to the medicine, but they also provide data about safety and efficacy on an ongoing basis. That may, of course, depend on the sponsor, the resources they have, and even the clinical trial centre being able to do that. So some other matters need to understand, of course, the differences between illicit cannabis and medical cannabis, and of course, THC content for investigational products and driving is a really important theme. I know great work that's been conducted at the Lambert has certainly shown that, you know, there's good evidence now that CBD-only products really aren't a major contributor. THC is a bit more complicated, but certainly some parts of those studies show that at lower doses, it might even improve driving ability. And it's been a very interesting area. There's more work needed here. And at the moment, the legislation, you know, is very strict about having THC in your system and driving. And of course, remembering that the pharmacokinetics of tetrahydrocannabinol is actually, given the very lipophilic nature, it stays in the system for a long, long time. So that's an important thing to appreciate. We also have obviously very strict regulations, depending on whether it's a Schedule 8 or Schedule 4 medicine, or of course, soon Schedule 3, when those trials are available. The regulations for storage supply of medicinal cannabis products is very important. So working with your clinical trials pharmacy or a supplier in that regard becomes very important. I won't talk about IBs because that'll be picked up in the next presentation. So one of the things I'd like to bring you back to is this concept of a PICO. So PICO, of course, is how we might frame our questions. So what is the patient population? What is the nature of the intervention, which is summarizing some of the things I said before? What will you compare this to? And what are the outcomes that you'll measure? So you can see also the type of question you're asking and the type of study design. So PICO really, and you can see from the questions I've put there, which group of patients will be studied? You know, what are the main, the treatment, the dose form, and that includes, you know, which cannabinoids, what dose regimen, what dose form, what route of administration. What are the main comparators that you'll compare it to for this group? So who are the, you know, from that patient population, what is the standard treatment that you're comparing it to? And what will be the outcomes that you'll measure? And of course, measuring efficacy, does it work, is very important. That's beneficence, but also measuring non-maleficence, possible adverse effects, is something that every protocol should include, usually as a secondary aim, unless it's a safety study in phase 4. And ethics committees will really push you to make sure that there's appropriate monitoring for that. And of course, it does come down to all those things we know about epidemiology and design of studies. Should it be blinded? You know, do we randomize treatment? Is this open label? How do we do that? And justifying that design becomes very important. There is also a unique aspect around capturing inclusion criteria, exclusion criteria, and even what we monitor when we think about all the factors that might be influencing the response of a person to medical cannabis. And I think this particular graphic summarizes some of those things, whether it's about other medicines, their previous exposure to cannabis, and we're thinking here about inclusion criteria. I've mentioned the route of administration, dose. Also that idea of chemovars, the idea of the possible entourage effect, where there may be cannabinoids of interest that the dose form is based on, but they could include within that extract other cannabinoids. And there's been some great work at the Lambert to look particularly at that. CBD content I've got, and possible drug interaction. So there's a bit of a summary of those factors that need to be importantly considered in designing the study, but also included in the protocol as well. So maybe just to finish up, ethical considerations in designing trials. So I think I've emphasized a few times now which product and its characteristic, justifying the dose regimen, the route of administration, and the dose form itself. What is the indication in that PICO way of thinking? What is the comparator? And of course, the quality of that product becomes very important. Evidence of GMP, certainly when it's needed in phase 2, 3, and 4 study. So generating evidence for safety and efficacy is product specific, particularly in the medical cannabis area. So that idea of PICO and study design is very important. So I'll finish there. Obviously, happy to take any questions later on, and I'll hand over to Anastasia for her presentation. Thank you.
Thanks, Andrew. I just want to encourage people to use the chat function to capture your questions, and we can go through all those. And now we have Anastasia's discussion. Thanks, Anastasia.
Thank you very much. I'm very excited to be here today, and fantastic talk from Andrew. That was a great overview of this current topic, and I will provide a few more practical considerations about designing cannabis-based medicine trials and how to best engage with HRECs. So just as an introduction, I'm a research fellow at the Lambert Initiative, and I'm also a recent PhD candidate that just completed a trial looking at the effects of a cannabis-based medicine in chronic insomnia disorder. And so I hopefully can impart some knowledge onto you today on the best way to engage with ethics committees. So these are some very helpful resources. I know Andrew has already touched on some of these, but they will be very important to you to refer to when preparing your ethics application, and good starting points to getting your head around what's required. And I'm sure these are all very, for many of you, are very familiar. So I'll leave those there. So just to provide an overview similar to what Andrew covered, as you all know, cannabis has been used for millennia. It's currently, the uptake of it and the interest of it is rising in Australia and overseas, and there's a strong consumer demand. However, we're in a situation where the cart is before the horse, where medicinal cannabis prescription rates are going up. However, we have limited data to support the therapeutic claims and safety for a lot of these indications. So for example, with PTSD, publications of my colleagues at the Lambert Initiative, showing the rise of prescriptions for various indications. And so really the evidence is still catching up. So what the ethics committees want is a risk-benefit analysis for the therapeutic indication that is being proposed. So really it's important to cover the clinical need in this indication, the prevalence, how many people are affected by this condition, and also the failure of treatment of current standard treatments. So that's kind of what Andrew was referring to, is that medicinal cannabis is not a first-line treatment, and a lot of the time, or most of the time, it is a sort of an add-on treatment. So it will be looking as an adjunctive treatment to current standard treatment. So this is something to consider when writing up that ethics application. There's obvious concerns about THC-based medicines, so driving employment, and also the next day cognitive effects, which we're exploring in a lot of depth at the Lambert Initiative. Post-trial access, which Andrew touched on. So of course, if, for example, high-dose CBD works for an indication in a clinical trial, the patient may not be able to afford that if they were to access it through special access schemes. And I'll also touch on specific requests that HRECs often provide in terms of advertising and recruitment for cannabis-based trials. And I'll go into a little bit more detail later on. So again, won't go into too much detail about the different phases of a clinical trial. However, I do want to just point out that majority of clinical trials looking at cannabis-based medicines are sort of in that phase 1 to phase 2 kind of region, where we're trying to identify safety and tolerability, and also identify the optimal dose, usually in a small number of people, usually between 20 and 40. And this will help us to optimize the design of subsequent pivotal trials, looking at phase 2, phase 3, and then obviously into post-marketing. So I'll touch on trial design first. So as I said, the ethics committee would like to see an evidence-based rationale, and so a justification and a risk assessment supporting the decision to pursue cannabinoids for that indication. And I want to again bring up PICO, which Andrew went through in much more detail, which is really helpful to kind of help you design that justification. Trial design selection as well is important. So obviously you've got the different types from open label to randomized controlled trials. But you've also got the different types of ways of administering the treatment. So usually trials comparing to a placebo or an active control, such as a standard treatment, they would either use a parallel group or a crossover experimental design. And in the former, each participant receives only one available treatment, whereas in the latter, they receive both in random order. And what we're finding is that, well, we know that CBD and THC are highly lipophilic, and we know that they have a long window of detection in plasma. And so a washout period is typically designed, so specifically for crossover designs, where there needs to be a washout period in between each treatment arm. A washout period is generally only considered adequate if it allows one treatment to clear completely from usually blood before the next treatment is administered, so to rule out carryover effects. And what we're finding is that CBD and THC are actually lingering in plasma longer than anticipated. And we're about to publish a study which has collated evidence from 3 different clinical trials where a dose of CBD was administered, and we're looking at plasma to see how long it took for the CBD to wash out. And we're finding that, again, it's, as I mentioned, it's taking longer to wash out as we expected. And so crossover studies are to be conducted with caution, particularly with higher doses, and particularly with repeated dosing regimens. And so this suggests that potentially parallel groups might be more suitable, but in some cases, crossover designs are unavoidable, and certainly happy to discuss kind of the issues relating to trial design after the talk. So in terms of the investigator's brochure, this is to help you define and characterize the investigational product. So it's a document that basically collates all the information about the quality, safety, and effectiveness of an unapproved IP or investigational product. And you do this with summarizing the relevant clinical and non-clinical data. It's typically prepared and maintained by the manufacturer, but it is the sponsor's responsibility to prepare or have the IB available for the ethics application. And I refer to the ICH guideline for good clinical practice. Everything you need to know about the IB is in Chapter 7, and it includes a step-by-step process of what needs to be included. And so the main point here is that there needs to be known stable active components so that the treatment outcomes can be compared and replicated. So in terms of that, I refer to the TGO 93 order, which I know many of you will be familiar with, and it just specifies what an active ingredient is. And this may become relevant for investigational products that are botanical extracts that may contain lots of different chemicals and ingredients. And so this is what the TGO 93 defines as an active ingredient. So THC, including any corresponding acids, greater than 1%, and then any other cannabinoids greater than 2%. So the stated content of the active ingredient is what's specified on the label or has been approved for the purpose of a clinical trial. And there's also a range of the stated content. So obviously it's not, you have to ensure that it is within that range. And so here is straight out of the TGO 93, the different dosage forms and the range of stated contents. And this is based on the average content of the active ingredient, and different measurements apply for the different types of dosage forms. So this is something to keep in mind, and obviously keeping in mind the different types of products that can be included in cannabis-based medicine trials, which I'll go into next. So selecting a cannabis medicine. So as Andrew mentioned, cannabis medicines come in all different types. So some may be highly purified, some may be isolate, some may be whole plant, and also the route of administration changes. So it could be vaporized, orally administered, sublingual, topical, etc. So the ethics committee wants you to see a justification for the dose and the route. And as Andrew mentioned, the pharmacokinetics and the pharmacodynamics is important, and you have to be able to explain the reason for using these types of medicines and that specific route specific to that indication. So for example, you might choose an oral product that is absorbed more slowly and has a longer duration of effects, which may be suitable for treating, for example, insomnia disorder, because we know that the drug will last longer overnight in order to assist with sleep. if that's something that is being explored in the clinical trial. And obviously it has to meet good manufacturing practice and also be in line with federal and state regulations. So refer to TGO 93 for the federal regulations. In terms of accurate dosing and administration, what you can do is use a broad range of published data to inform the dosing schedule. You can infer from preclinical studies, data from clinical populations from other than your indication, and also from other cannabis medicine trials. So, for example, if you were to use a 20:1 CBD to THC product, although there's been very few trials out there using that particular ratio of cannabinoids, you may refer to the Sativex and the Biximol trials with a 1:1 ratio of CBD to THC, showing at least in that ratio the safety and efficacy with that product. Not to say that it's a good replacement for the evidence with a 20:1, but at least you can infer from other previous trials and provide at least some measure of safety with that product. And obviously consider factors that also may affect absorption. So as I mentioned, route of administration, but also things such as cannabis use history. So the participant's previous exposure to cannabis can affect their experience as objective drug effects to that particular dose that you are intending to use. And also body weight and fat composition can also affect the rate of absorption of a cannabis product. Other things to keep in mind with the investigational product is storage and dispensing. You have to be familiar with the scheduling of the medicine and the different regulations around such products. So Schedule 8 products typically require much more stringent storage requirements, as well as specific features, and that applies to all medicines in clinical trials, so child-resistant caps, instructions for storage and use, etc. So I know this will be all very familiar to most of you, but important to touch on. Moving on to participant safety. Patient selection and safety. So THC containing products are generally not appropriate for certain patient populations. So pregnancy, current psychotic disorder, and also unstable cardiovascular disease. Obviously important to note all adverse events, and given the risk of drug-drug interactions with both CBD and THC, important to include blood tests for any treatment emergent abnormalities. So this could be monitoring that's inbuilt into the trial protocol, where liver function tests are conducted over certain intervals, or perhaps at baseline to ensure there's no liver impairment at the start of the trial before they initiate dosing. And obviously driving is one of the important factors for THC-based medications. Important to exclude participants who are undergo mandatory drug tests for either their employment or court order. And also important to include explicit warnings about driving and the legal ramifications about using THC-based medicines. And this should be in all the patient-facing documentation. And often in trials, we do provide patients with taxi vouchers or other reimbursements to help them with travel. Drug-drug interactions, obviously, as I mentioned, important. Cannabinoids metabolized by the CYP450 system and enzyme system. And so that's a very common site for many drug-drug interactions. And so recording all the co-administered medications, including supplements, their alcohol and illicit drug use intake is important. And obviously instructing participants to avoid use of external cannabis and illicit drugs and certain medications if applicable throughout the duration of the trial. So you can confirm abstinence to external cannabis use using commercially available urinary drug screens. So moving on to informed consent. As Andrew mentioned, setting realistic expectations given the very increasing interest in cannabis-based medicines, and obviously the attraction to very vulnerable patient populations who've tried everything and are very desperate. So it's important not to make any claims about safety and efficacy. And I always like to share the ethics-approved participant information sheet very early to allow them access to discuss, allow them time to discuss it with their GP, with a trusted family or friend prior to consenting to the trial. And as Andrew mentioned, discussing that post-trial access is very important. So, and that needs to be clearly communicated in the information sheet as well, where that might be an open label extension arm, or that the medication supply will just simply not be available after the trial, or that a same or similar product is available through the special access schemes, but also important to note the high cost associated with this particular pathway. Now, finally, I just want to touch on advertising and recruitment. So as you know, cannabis-based medicines are unapproved therapeutic goods, and they are prohibited from being advertised to the public. Human research ethics committees are very particular about how these types of trials are advertised to the public. So whether that might be through traditional media or electronic media. Generally speaking, we do not use the cannabis imagery, nor do we make claims about safety or efficacy, but that's pretty straightforward. And so typically the way that we promote it is through a simple line of saying, new study inviting participants aged XYZ to try a new medication to treat the indication. So it's quite vague, it's very general, but it allows individuals to look specifically only at the eligibility criteria and think of it from that point of view, as opposed to what is the treatment. Obviously, once they make contact, you provide them with more information about what the treatment is, but it does avoid recruiting a biased sample. So people who already believe cannabis is good, and it potentially could inflate the placebo effect, or you're inviting very vulnerable individuals who are seeking a treatment and are desperate to try anything, and may not necessarily have are giving informed consent given their situation. And obviously providing balanced statements outlining the lack of clinical evidence and the need for more safety data. So I point to the advertising guidance. This is more generally for businesses involved with medicinal cannabis products, but the HREC committee do also guide us towards this document as well, just to look at how this might apply to the way that we advertise to the public about clinical research trials. So I will leave it at that, and I'm happy to take any questions.
Okay, thank you very much, Anastasia. And we do have some questions coming in through the chat. And Andrew, if you want to join us again for questions. Anastasia, I do know that you're on a time limit, so if people have questions, particularly for Anastasia, please get them in as soon as possible so that we can grab her experience. Just to go through a couple that have come through the chat, what's your thoughts when it comes to planning or conducting the trials with regards to addressing placebos, particularly given the different and distinctive smell?
Maybe I'll start with a general comment, and then I'll throw to Anastasia for practical tips. Obviously, a placebo-controlled trial, it becomes a very important consideration when you're trying to answer a clinical question and even a question about safety. So if you're trying to decide if the addition of a medical cannabis product to a current treatment regimen, reduces seizure frequency or chemotherapy-induced nausea and vomiting, then it becomes very important to have a placebo. And it's been a really difficult thing to match. Of course, a placebo, by definition, should look identical to the active treatment and really trick both investigator and the participants. So sometimes if it's and they can be very expensive to access, they need to be prepared under GMP conditions. So this is a incredibly vexing challenge for all sorts of areas, not just medical cannabis. But placebo is highly desirable. Over-encapsulation has been one approach, but it's not a trivial thing, because that may even impact on bioavailability. So we need to be a bit careful there. Sometimes also they might use, if it's an oil, they might use a similar base, such as, mid-chain triglycerides, to have a similar taste and smell, but it's not always easy. But Anastasia, any advice around placebos and the general approach?
Thanks, Andrew. Yeah, I echo your sentiment. It is, cannabinoids are notoriously difficult to blind, especially if you've got any THC component in your medication. And certainly over-encapsulation is one option where obviously you're trying to make the visual appearance the same, the taste and smell the same. However, sometimes you can't achieve that. So generally speaking, oils, what we have done in the past is using a masking agent. So again, you do not want to tamper with the actual medication because that has to be GMP manufactured. So any masking agent has to be external of your product. And so things such as mints, very strong spearmint, things like that, lozenges can be used. But again, this is only in a setting where it's a controlled setting where participants are coming in, you can observe them. In a setting where they have to take the medication home and take it over a period of time, blinding makes it even more harder. So you do have to think very carefully about things such as this. And certainly finding excipients that match in color with the active medication is important, using amber vials, things like that, to make sure that the bottles don't look different, so you can't actually see the differences in the liquid. But certainly encapsulation is probably the best way to hide it. And certainly, I guess, yeah, in a controlled setting where you're administering it acutely, there are certain things you can do, such as masking agents. But other than that, it's certainly a tricky thing to manage.
Look, sometimes too, if the endpoints of the study can be objectively assessed, and they're less likely to be influenced by what people think they're taking, but then maybe you can get away without using a placebo. But an ethics committee will probably ask that really question: justify why you are using a placebo, or why you are not using a placebo to address your question, because that would be seen as the optimal scientific design. Logistically, becomes very difficult, in accessing a placebo. That's the reality. Yeah.
All right, thank you. That's excellent. Another question from our group: if trial material contains cannabinoids other than THC or CBD, is it anticipated that more safety and mechanism data would need to be provided about that specific cannabinoid? So currently, majority of cannabis products are able to use literature that supports safety of THC and CBD, but will this be enough for other cannabinoids?
I'm going to throw to Anastasia, who I know is probably working on this very challenge given the entourage effect research that's happening at Lambert. But do you want to comment first, Anastasia?
Sure. Yes, that's right. Yeah. So with I think the first thing you want to confirm is whether the other cannabinoids that are present in that formulation are active ingredients as considered by the TGO93 definition. And if they are over the 2% concentration, then technically you would need to provide evidence to explain, to justify using that particular formulation for that indication. And we have looked at other cannabinoids other than THC and CBD in our research, and we have leaned on preclinical research to help explore, I guess, safety from in a non-human trial. But certainly you can refer to the fact that a lot of whole plant cannabis and vaporized cannabis does contain minor components of these cannabinoids. And so these have been ingested by humans over time, and they have been ingested in a clinical setting or in a clinical trial setting. So I guess it just depends if the dose of those other cannabinoids are exceptionally high. So if you're looking at dosing, 300 milligrams of CBN, you may need to provide a justification of why you're going that high and whether there is any safety data for that particular dose. So it's really just about a balancing act between those two different factors.
And why, I suppose getting advice as you plan those from, advisory board or clinical advisory board could be of value. The, part of the summary there too is that the threshold of whether those components, are considered an active ingredient. Of course, if you're making a claim, that your product might have benefits, then an ethics committee would say, okay, so it has added benefits, it may have added potential harmful effects. So it is about presenting a cogent scientific argument with all the information you can get, preclinical, obviously human use experience with that, and also understanding from a quality point of view exactly how much of those cannabinoids are there, and you know, at what threshold. It becomes very important. So it is about generating a case around the balance between possible benefit and harm.
Thank you for that. Sort of going on from that, what's your experience or thoughts on presenting a very literature-based submission to the HREC for things like the non-clinical evidence for a product, rather than the far more traditional product-specific data built through the non-clinical and then the early stage clinical trials?
Do you want to go first from an IB point of view, Anastasia?
Sure. From an IB point of view, you have to be quite explicit when you say that this particular product has not been tested. If it's a custom-made product that has never been, I guess, tried in humans, you do need to be explicit in saying that this particular formulation has not been tested in humans. However, you then provide supplementary evidence from other trials, from other indications where a similar ratio of cannabinoids or a similar formulation has been used previously, at least to infer some level of safety.
And I think, so that's a critical part. I mean, the literature clearly has a very important role in providing, you know, the justification and rationale for a particular study. So robust literature contribution is very important. Obviously, you don't write all about CBD and then your product has CBD and THC. You've got to provide the relevant literature for the nature of the product that you're looking to investigate. I would add, if there's similar dose forms, you know, that have been studied, much of the evidence, you know, is product and combination specific primarily, because they have different, you know, I would say pharmacokinetic profiles. So some of the studies that I've seen actually did a preliminary study comparing their novel formulation with Sativex, so a product that's on the market, and demonstrated that the expected concentration time profile of CBD was similar to what you'd expect for Sativex. So that was a nice way to link the evidence and safety for that type of exposure. Obviously, it's a different product, but to say it's in the ballpark of a similar exposure, we mean concentrations in the blood, which gave the committee some confidence around the idea that, okay, well, this isn't going to be excessively high or perhaps even excessively low. And that's part of linking the evidence in the literature to the nature of the product. It's not a direct link, but it's a step towards it. So you can see how you want to use the relevant literature to make the case to support, you know, what we know about what we could expect, for this new product that's being tested.
Oh, excellent, thank you. What are your thoughts with regards to having your proposed protocol independently peer-reviewed prior to submitting it to the HREC, and sort of incorporating that peer review response, or how you've responded to peer review response as part of your submission?
Maybe just from a HREC point of view, Anastasia, can I go first? Is that right? So most HREC applications will have a, they ask you, has your protocol been peer reviewed? And if you think about what the HREC is after, there is, what's been the process of how this protocol has been developed? Is it based on, as we say, a sound understanding of the literature, critical appraisal? How have you generated the research question? Obviously, the PICO approach that we both mentioned becomes very important there. And of course, the peer review discussion, sharing it with clinicians, the area, pharmaceutical scientists, you know, people with experience in prescribing and using cannabis-based medicines. What that does also for the researchers is help them think about all the things we're talking about today and make sure that they've covered the field. The design is relevant, the population group is relevant, how they'll recruit, how they'll advertise, the inclusion, exclusion criteria. It's also, it means that the ethics committee can have some confidence that what they're being presented with is a robust document that's been through the ringer, if you like, of being investigated. Now, that can be done commercially in confidence. So many of those peer review processes are conducted in confidence, and people do provide feedback there. It's not alternative to HREC approval, but I think from an investigator's point of view, it gives you a good sense. You're very committed to the project. You'd like to see it successfully conducted, this study. So if you've worked on it very closely, this has been my own experience in developing protocols, you know everything about it. So when you share it with colleagues in a peer review sense, who are independent of the project, they will always add value to the quality of the final submission. So that'd be my take. But Anastasia?
I think Andrew's covered that perfectly. Nothing to add.
Okay. And one final question here is, do you have any tips on engaging with the HREC? I'd assume this is midway through the process while you're in discussions, where you disagree with the position that the HREC has taken.
Certainly, yeah. So in that instance, I think you have to have, again, a good justification and reason as to why you are disagreeing, and also providing evidence or examples as to why whatever they're recommending is not appropriate for your trial. I think it's really just about putting together a solid argument. Any tips, Andrew?
Yeah, absolutely spot on. I think, look, even as I'm a member of an HREC, I'm an investigator, you know, the comments come back from the ethics committee, and, you know, there's always a moment of, it's like when the comments come back from a manuscript, you're thinking, they really don't get it, do they? But actually, I suppose I see either side of it. And HREC is using the national statement to ask questions of the investigator to clarify aspects to make sure that it meets that national standard that is set there. And a good ethics committee will always link a question to part of the national statement. You know, according to national statement 3.1, please provide more information about this. So it's not just random forms of torture that an ethics committee would like to be involved in. Actually, they're trying to clarify to get to the point of making a decision about the ethical design, conduct, and, you know, implementation of this particular study. So I would, my general approach, take each of their comments, put them in a table, respond to each of them. Because one of the things as a reviewer, if I'm struggling through a document trying to find how they responded and where they responded, very, very difficult. Make track changes on your protocol and your participant information sheet and submit those so the changes are really obvious. You know, where there is we agree, make the change. Where there is you disagree, provide evidence, as Anastasia said, as to why that is either not appropriate or not possible. And it is, it's not inappropriate to say, you know, our funding doesn't extend to that, or, you know, this is not the priority of this study. We're going to look at that in a subsequent study. Look, there are ways to, you know, take on the questions. I mean, my general sense, though, is that the ethics committee I'm involved with and have been involved in previously when I was at Concord Hospital, we work very hard to only ask those questions that aren't just about correct the spelling. That's up to an individual. It's about, you know, decision points about whether or not, you know, this meets the standard for the national statement. So, you know, respond. I'd like you to rethink about those questions as ways to improve your protocol to make sure that the study is optimally designed. And I'll tell you now, it usually improves the likelihood of getting it published once the study, you know, is complete, because it'll be a high quality study.
Brilliant. Thank you for that. Are there any last minute questions to come through chat? We seem to have got through that and covered a lot of ground. Looks like we've got most of them. I would like to thank, on behalf of MCIA's research working group in particular, Anastasia and Andrew for your participation and for bringing your knowledge to this group and sharing it. Thank you, Andrew, too, for saying that you were willing to take on sort of additional questions. Sure. If people think of them later. And yeah, I think we've covered everything, and I wish to thank you again for your brilliant presentations, both of you.
Thanks, Anastasia.
Thanks, Andrew. Melinda, thanks for having me here.
No problems. Thanks, everyone, for your questions and your engagement.